Hi, I'm Anna Kuchina. I'm an assistant professor at Institute for Systems Biology, where we study bacterial physiology and bacterial behavior one cell at a time. And so today we'll be discussing the paper that we just published in Nature Communications, where we looked at the interactions of a gut microbe, Bacteroides fragilis, with the bacteriophage, or the virus that infects it and kills it, that we isolated from Seattle municipal wastewater. And so in this paper, we study this pathogen because it's normally a commensal gut microbe that is generally harmless, but sometimes it turns into a pathogen, for example, when the intestinal wall is breached and it escapes into the bloodstream and causes infections. And so we were interested in how these bacteria interact with the phage as in the process of the phage infecting them and killing them, using our technology that we developed for studying gene expression in tens of thousands of microbial cells at a time. The lead authors of this study are Anika Gupta, the molecular engineering graduate student at University of Washington, and Dmitry Sutormin, a postdoc in my group. This is also a collaborative study with Neil Day at Fred Hutchinson Cancer Center and his postdoc Norma Morella, which is also a co-lead author of the study. So, Anika, what can you tell us that we found when we looked at the individual cells of Bacteroides fragilis, this bacterium, and it was challenged with the phage?
Yeah, so when we looked at individual B. fragilis cells exposed to phage, we could really see these infected cells in which the virus was replicating. And we were also able to computationally reconstruct the different stages of phage infection and see really how are these bacterial and viral expression states changing over time. And interestingly, we were also able to detect a fraction of cells that were not actually infected by phage. And that kind of tells us that within this phage-treated population, not all cells are equally susceptible to phage.
Well, this is surprising, right? Because all of the cells are supposed to be genetically the same, because they're descendants of the same cell that we grew in the lab. But when we challenged it with phage, then we saw that a fraction of cells actually remained uninfected. So Dmitry, maybe you can comment on how did the cells protect themselves? What did we find?
Yeah, indeed. And one of the strongest signals we saw in our data when we compared infected and uninfected cells, we observed a specific selection of certain genes related to the capsular biosynthesis. And it turned out that some of these capsules, they are protective, so they allow bacteria to survive during the phage infection. But other capsules are sensitive, so almost all the cells having these sensitive capsules, they were heavily infected with a phage, and they are going to die because of this infection. But that's not the only thing we observed, actually. And interestingly, it turned out that the Bacteroides fragilis, it has a very plastic genome. It means that it is exposed to multiple chromosomal rearrangements. A lot of recombination is happening here. And other genetic losses, they are also contributing to the phage infection or a phage protection. Among the genes, we outlined a couple very novel and interesting candidates. For example, several clusters related to the fimbria biosynthesis. The fimbria are bacterial appendages, build of proteins. They may be used for adhesion to biofilm formation, so it's work in progress. We don't really understand now what's the mechanical basis for that, but we just picked up these candidates and observed that they contribute into the protection of the population.
Thanks, Dmitry. And so it's also well known, isn't it, that bacteria actually have immunity systems, so to speak, or defense systems that specifically protect them from viruses such as phages. So maybe, Anika, you can comment what did we find about the involvement of these immune systems in protecting the cells from the phage?
Yeah, so these bacterial defense systems also did seem to play a role in protection against phage. We observed that in individual cells, the expression of defense systems was very variable. And in certain cases, with some defense systems, we observed that they were able to increase phage protection when expressed alongside one of those protective capsular polysaccharides.
Oh, that's fascinating. Thank you. So I guess to wrap up, maybe Dmitry, you can comment, what are the implications of the study and what are the next steps for us to applying our high-throughput technologies for studying bacteriophage interactions?
Mm-hmm. Thank you, Anna. So for me, this is, first of all, a very nice biological story that highlights how adaptive and plastic the bacterial systems are. We used to think that bacteria and viruses, they're kind of robots that have some hardwired genetic circuits and they respond to specific stimuli in a specific way. But this study shows that there's a highly multi-layered system, and bacteria can respond to the viral infection in multiple ways, and all these layers, they interact to each other. That is a capsular polysaccharide layer, that is a fimbrial layer, that is defense system layers. They all interacted. And I think that's not the end of the story, actually. A small cliffhanger here. We recently touched another mechanism here, so stay tuned, and maybe we will see another player in this playground. If speaking of a broader perspective, how it is related to the phage therapy, because the phage therapy is an evolving field, and it's a very nice alternative to the antibiotics because it's more precise and it can handle pathogens which are not susceptible to antibiotics. But imagine if you have a pathogen of a similar plasticity, of a similar adaptability. That's a huge challenge for the phage therapy because bacteria may have multiple ways to escape this type of treatment. And our single-cell studies, they paved the way to elucidate all these multiple mechanisms in nature and how these mechanisms are interacting to each other. So potentially using single-cell transcriptomics, we can find a specific type of treatment, a specific composition of a phage cocktail which will cover the entire adaptive landscape of bacteria and which didn't give them a chance to escape from our treatment.
Thanks, Dmitry. This is fascinating again, and the phage therapy angle is really an unexpected angle for us because we are mostly interested in very basic questions such as what are the potential implications of bacterial variability in terms of expressing different genes in different parts of the bacterial populations to bacterial lifestyles. But now, given that we've used our technology, single-cell transcriptomics, to profile interactions between the bacterium and the phage at a single time point, but we were able to, using this snapshot, reconstruct the full cycle of bacterial infection and also find the cells that were protected. Again, from a single snapshot, gives us now the opportunity to extend our studies to studying these interactions in the context of the microbiome itself, right, without potentially culturing or isolating the phage and the host pair. And so this potential that we sort of unlocked with this study, and we demonstrate how we use our technology, microSPLiT, to do this, this potentially can open the door in profiling and solving one of the big challenges of studying microbiome phage interactions, because it is actually very hard, a very hard challenge to isolate the bacterium, the host bacterium, and the phage sometimes, right, to study their interactions in vitro in the lab. But if we can just take the microbiome sample and take it as is without any transformations or culturing and then apply our technology, we will be able to, similarly as described in this paper, computationally reconstruct these processes. Thanks, and congratulations on the paper. Please check it out. It will be open access in Nature Communications.